right, so we're starting the accessory organs, Roman numeral 5 in your outline, starting with the liver. So the liver does a lot of things for your body, a lot of functions here. I only listed, this is a shortened list. We know that the liver detoxifies substances from our blood, so our liver is taxed with getting rid of all the junk we take in, all the pesticides and herbicides that we're accidentally eating on our foods, things that we inhale, eventually get into our bloodstream. So any sort of prescription drugs that you're taking, everything's detoxified in the liver. The liver also synthesizes bile. So bile here, it emulsifies lipids in the duodenum. So emulsify, I just want to make sure you guys know the word emulsify. It's to break large droplets down into smaller droplets to increase surface area for enzymes to break the fat down. So it makes it easier for the enzymes to access the fat and to be more efficient at breaking down the fat. So bile doesn't it doesn't enzymatically break down fat, right? Bile is not an enzyme, but bile helps to create smaller spheres so that the enzymes can work on more surfaces. All right, so hopefully that makes sense. It makes the bile is then stored in the gallbladder. So the gallbladder is a separate organ. It's attached to the inferior surface of the liver, but it stores bile. So the gallbladder doesn't make anything. It's just a storage. It's the liver that creates the bile. The liver also destroys white blood cells and red blood cells. Remember, the spleen also has that role. However, if you get your spleen removed, the liver can take over for all that. The old heme in your red blood cells is converted to bilirubin. Then bilirubin is excreted in the bile. And this is the substance that makes the bile green. So if you do have a cadaver dissection in your life where you see images of the gallbladder, it'll be green because bile is literally green. It's a storage place for glycogen. It also stores lipids, lots of iron and copper and fat. It makes plasma proteins, namely albumin. It makes clotting factors. It makes complement. So it does a lot for your immune system. Fluid balance, the albumin there maintains osmotic pressure in your blood. It metabolizes carbohydrates. It metabolizes proteins. It does a lot of roles for digestion, but also other. So it kind of overlaps with other things, but primarily it does a lot for digestive system. So let's take a look at the anatomy of the liver. Your liver has, believe it or not, 4 lobes. There's 2 lobes that are very obvious here. The right lobe on the right, the left lobe on the left, divided by the falciform ligament. Now be careful because the superior surface of the liver is called the coronary ligament, and the coronary ligament comes here. Then there's a deeper fissure where it becomes the falciform ligament. The falciform ligament then wraps around to the posterior surface. It's kind of cut here, cut away, but in the back it's going to be called the round ligament. Okay, so same thing, 3 terms based on where it is: coronary, falciform, and round. On the inferior surface, we have 2 smaller lobes. So we can see the quadrate lobe, a little small quadrant next to our gallbladder, and then we have a separate caudate lobe, right? Caudate lobe here as well. So the quadrate and caudate lobes are on the inferior surface of your liver. We can see that, let's see, this our round ligament would be continuing up here, okay? So if we had models that you needed to study in class, you would have to flip the model upside down to see the round ligament. So now let's look at the Porta Hepatis in your notes. So the Porta Hepatis is this region here, right? We have a big bolded word here, and there's 3 vessels or structures that are going to enter and exit in this area. So Porta Hepatis means the port to the liver. So it's a major entrance and exit for these structures. So we have the hepatic portal vein. This should be very familiar from the vasculature chapter, right? This portal vein is bringing blood. It's bringing blood from our digestive tract, right? Remember that there was the gastric vein, the superior and inferior mesenteric vein, as well as the splenic vein, all coming and converging at the hepatic portal vein, coming into the liver so the liver can detoxify the nutrients in the food before it enters general circulation. So the hepatic portal vein is here, the entrance. We also have the hepatic artery proper, and that's just delivering oxygenated blood to the liver. And then we have the common bile duct. So remember that the liver is creating bile, and the bile exits the liver through this structure here. Okay, so that entire region is called the Porta Hepatis. Now, let's look at the internal anatomy. Internally, the liver is going to look like little hexagons. So the hepatocytes, the cells of the liver, are arranged in hexagonal rows, and these are called lobules. So you see there's a connective tissue called a septum between the lobules, so that's why it's called interlobular septum. But it's going to create this organized feature, a hexagon. And if we were in class, we would draw this in class, but we aren't in class right now, so just pay attention and maybe you can sketch out an image. It would look like this, what we would draw in class. It's a closer up look at the microanatomy of the liver. But let's just take a look at this picture before we move on to the other picture. All right, so what we see here are a couple lobules, and in the center of every lobule there's a central vein. Okay, so we can see the colored in central vein here. We kind of removed the blue blood here, but there's our central vein. And then running between this hepatocyte, so everything that's yellow here are hepatocytes or liver cells. The groove between them, those are called sinusoids, okay? So the sinusoids are where blood is going to travel through. Inside the sinusoids, we're going to have Kupffer cells. And then there's another structure called Bile Canaliculi that you can see right here. And I'm going to mention this again in the next picture. There are 6 corners of the hexagon. So let's take a look here, right? And then let's take a look over here. You have a repeated 3 vessels at each corner of the hexagon. So these are the same 3 structures that are at the Porta Hepatis. There is a branch of the portal vein, so the big blue guy is the hepatic portal vein, branching, and each branch will follow the sort of point of the hexagon. We're going to have a branch of the hepatic artery, and we're going to have a branch of a bile duct. Okay, so every corner is going to have those 3. So those 3 are called the portal triad. Okay, so the portal triad contains a branch of the portal vein, a branch of the bile duct, and a branch of the hepatic artery. All right, so let's take a look. So I want you to notice how the blood, right, is traveling through the sinusoids. So looking at the sort of blow-up picture, we have the portal vein bringing blood that is rich in nutrients, right, because it's coming from the digestive system. But as well as being rich in nutrients, it's also rich in potential hazards, right? It could have chemicals that are nasty to your body, and your liver is going to detoxify them. So this is going to run through a sinusoid, and within the sinusoid you're going to have these Kupffer cells, so these pink guys. Those are resident macrophages that are there to phagocytize any pathogen or potential debris that comes into the liver from your intestines or from your stomach or from your spleen. So those Kupffer cells are residents of the sinusoids. Then you see a sinusoid, so the blood is going to come in, it's going to move through the sinusoid, past our Kupffer cells, through the sinusoids, and then eventually dump into the central vein, right? So remember, this is in the middle of our hexagon. The oxygen-rich blood coming from our hepatic artery is also going to run through the sinusoid, and it's going to deliver oxygen to the cells, right? So it's going to just diffuse the oxygen, and then also that blood will travel into the central vein. So the blood comes from each portal triad runs through the sinusoids, and then enters into the central vein. All right, so as it moves through the sinusoids, Kupffer cells can trap or phagocytose any debris or pathogens that are in the blood. Also, the nutrients can be packaged up. So the hepatocytes here do a job in metabolizing carbohydrates, metabolizing lipids, packaging your cholesterol. So there's a lot of activity that the hepatocytes do with your nutrients taken in. Now with the bile is going to run the opposite. You don't want bile and blood to mix, right? That's kind of a no-no. So the cells that create the bile, the hepatocytes are making the bile will go into these tiny canaliculi. The canaliculi are separate than the blood. The canaliculi is going to take that bile and then route it into the bile duct. Okay, so every corner of the hexagon, that lobule, will have a bile duct accepting the bile created from that lobule. All right, so blood moves towards the central vein. Bile moves backwards into that bile duct. Okay, and so that's basically it. You would draw this. I talk about that, and that's it. And then eventually, let's talk about what eventually happens to the blood. So the blood enters into the central vein. The central vein then moves out of the liver. So let me show you the picture here. Right, the central veins will converge into larger and larger veins and finally exit the liver through the hepatic veins. So this hepatic vein will take the blood from the left lobe and put it into the inferior vena cava, right? So there's a couple of hepatic veins left and right feeding into the inferior vena cava when the blood is in the inferior vena cava and it's on its way to the heart to go to the lungs and then the rest of your body. All right. So let's take a look at a histology slide. So the liver is part of your histology list. You're going to need to recognize liver and recognize a few things. So I'll list them off right now. You need to recognize liver, and you do this by looking at the organization of a lobule, right? I can make out this connective tissue here. I can kind of see that it's creating a hexagon, and I want you to know that that is a lobule, right? So the word lobule should be something that you can remember. So this is one unit, this is one lobule. Then we have a central vein in the center, right? You can see it's pretty obvious. Then we see the corner, right? So there should be a line here. Portal area should be going to this circle. So in the notes, it's called portal triad. However, your textbook also calls it portal area. So just recognize, right? These are portal areas. Here's a portal area. Here's a portal area. You don't need to recognize each individual structure. So I'm not going to ask you to recognize the portal vein or the hepatic artery or the bile duct. That's a little advanced. So just recognize portal area, hepatic lobule, central vein, and then call these cells that you see, all the pink cells, those are hepatocytes. And I also want you guys to know that the cracks you see how there are white, they look like cracks or fissures or spaces between the hepatocytes. You want to name those sinusoids, right? Because that's what they are, sinusoids. It's like sinusoid. All right, so that's it for the liver. So recognize liver and recognize those features that I just went through. This is another slide of liver. Okay, so you can see the lobules are pretty obvious. The central vein is pretty obvious. And then the portal areas or portal triads are just at the corners, all right? All right, so this is the complete-ish list of the major functions of the liver. Notice it says major. So there's lots and lots of jobs. You cannot live without a liver. The liver is very regenerative, probably because it's taxed throughout your whole life. And if it didn't regenerate, we would be dying pretty early. So your liver is very phenomenally regenerative. It bounces back from being harmed. But there is a point of no return. If you constantly harm your liver, you can get liver cirrhosis. Liver cirrhosis means that you need a new liver. You're harmed it beyond repair. All right, so this is the gallbladder, all right, located beneath the liver and stores bile. And you should know the pathways in this image. So you should know this image. You should know the gallbladder here. I didn't list the parts of the gallbladder, so that's not necessary. But I did list the cystic duct. So when the gallbladder is going to squeeze with smooth muscle, squeeze out the bile will enter into the cystic duct, okay, and travel through the cystic duct. Now, bile can also come from the liver. So the liver here is the right lobe of the liver, which is big, the left lobe of the liver, which is smaller. But you can see we have a right hepatic duct and a left hepatic duct. Those are the ducts coming from the liver itself. When they merge, we have the common hepatic duct, right? Remember that word common was the merging of two things, or before two things separated. Common hepatic duct. When the common hepatic duct and the cystic duct join together, then we have something called the common bile duct. Common bile duct. Okay, so you want to know all of that. The common bile duct will travel then all the way down and enter the duodenum at this little bump here. So remember the word for little bump in anatomy, that was called papilla. Papilla on your, or papillae, the plural form, on your tongue. Here's another papilla. So this little guy here, it's kind of like a donut. The center is open, it can be opened, it's hollow, and it can open to allow the bile to come into the duodenum. And so when the stomach releases chyme into the duodenum, the bile is also released, and so you can start to sort of emulsify the fats that you have just eaten. All right, so that is number 4 in your outline. I just went over all of the structures here. Now let's look at the pancreas. We'll come back to this in just a second. Let's talk about the pancreas first. So the pancreas is an organ, all right, accessory organ of the digestive system that is, looks like this, right? There's a rounded portion here called the head of the pancreas, and the head is going to be nestled in that curve of the duodenum. The body of the pancreas is this region here in the middle, and then the pointed end is called the tail. So it has a head, a body, and a tail. The pancreas has 2 ducts that runs through. So we have something called the pancreatic duct. So you can see this faint kind of golden color. This is all called the pancreatic duct. When you reach this point here, we can see that the pancreatic duct splits, and the pancreatic duct proper goes down and joins up with the common bile duct at that papilla, which we can't see right now. The upper branch of this is extra, and that's called the accessory pancreatic duct. Okay, there's another tiny hole that called a lesser papilla that I've not seen drawn in, but that's where more pancreatic juice can come in. So the pancreas creates pancreatic juice, okay, in terms of its job for the digestive system. So let's look at letter C in your outline, exocrine glands, and let's look at the slide that is the internal microanatomy of the pancreas. So the pancreas has 2 populations of cells because the pancreas has 2 different jobs. It makes pancreatic juice that helps to digest the food you eat or ate, and it makes hormones. So hormones, remember the word endocrine, endocrinologists, endocrine glands make hormones. Endocrinologists are doctors that study hormones and help you out with the hormone imbalances. But so the hormone producing cells are in little islands, or otherwise known as islets. Pancreatic islets are endocrine glands, and these make insulin and glucagon, 2 really important hormones that regulate your blood sugar throughout the day. Insulin, let's just talk about this since we're here. Insulin will reduce the blood sugar, so if you just ate food, the amount of sugar in your blood will increase because your digestive system is pulling and absorbing the nutrients into the bloodstream where it can travel to all the cells in the body. So insulin is critical in allowing your cells to accept glucose into the cells. So after you eat, your pancreas will produce insulin so that the sugar in your bloodstream can go inside your cells and be used. Glucagon is the opposite. It increases your blood sugar. So if you're starving and you need sugar in your bloodstream, glucagon will be released. It will signal to your liver because your liver stores glucose as glycogen. Remember the branched molecules of glucose. And so glucagon will break down glycogen. Glycogen will be broken down into those glucose molecules, go into your bloodstream, and then you have a sugar reserve that you're using at that point, right? So if you're really hungry right now, your body's probably making glucagon and you're breaking down your sugar reserves in the liver. So both of those hormones, and I'll introduce a word right now. When hormones do opposite things like glucagon and insulin, they're called antagonistic hormones. So they antagonize each other. They have opposite functions. And so glucagon and insulin are antagonistic hormones. They're made in different cells. We're not going to learn the cells inside the islets, but the islets are endocrine cells. The rest of the pancreas, which is the majority of the pancreas, are acinar cells. And the acinar cells are exocrine glands, so they're going to secrete their substances into a duct. And you can see the center, how it's hollow. You can see the duct, right? So the digestive enzymes are created here, secreted into the duct, that eventually becomes a pancreatic duct, and then leading into the duodenum. Okay, so let's look at what is in pancreatic juice. Pancreatic juice contains trypsinogen, which is an inactive form of this enzyme. It gets converted to trypsin, which is the active form, and you should know that digests protein. Lipases digest fats. Amylases digest starch. So this is the second place you see amylase. The first place we saw that was the saliva, and also lipase. Lipase is also produced in the saliva. So you see these enzymes again, and also all throughout the small intestine you see these enzymes secreted. Nuclease is secreted here in pancreatic juice. Nuclease breaks down DNA and RNA because everything we eat is a living thing, animal or plant, so it has DNA or RNA to break down. And then the last is sodium bicarbonate. So those of you who have taken chemistry, sodium bicarbonate is a baking soda basically, and it neutralizes. Baking soda is a basic compound, and it neutralizes the chyme coming from the stomach. So let's go back and just take a look at anatomically speaking, right? So there's our stomach. The stomach is churning the food with its gastric juice at a pH of 1 or 2, very, very acidic. The stomach can handle the acidity because there's a very thick mucus lining to protect the stomach wall itself. But once that chyme comes into the duodenum, we still have an acid. The chyme has a pH of 1 or 2, but the duodenum is not going to protect itself with thick mucus. So we have to neutralize that acid, and that's what the pancreatic juice does. That's what specifically the sodium bicarbonate in that pancreatic juice. It neutralizes the chyme coming from the stomach so that the duodenal wall, right, is going to be intact and not harmed. All right, so those are the substances in pancreatic juice. Definitely know those. So let's now go and look at this picture. So we can see that the pancreatic duct coming from the pancreas, the common bile duct bringing bile down. There is a control mechanism here. So there's a tiny sphincter called the hepatopancreatic sphincter. So you can find that word in the last section of this outline, the junction of the duodenum. So you have these terms here which are in this image. So here's our papilla, right? This is the bump you can see from the outside. Remember I said it was kind of like a donut with this hollow opening here. So this opens up to a wide area. This is called an ampulla, so the duodenal ampulla. This is where the bile and the pancreatic juice can converge before entering into the duodenum. The control, right? So we don't always want bile and pancreatic juice to spill in. We can control that. We don't, if you haven't eaten, there's no reason for bile or pancreatic juice to go into your small intestine. So the sphincter here is controlled hormonally. When your body has food in the stomach and small intestine, this will relax and allow the bile and pancreatic juice to enter. And when there's nothing there, it's going to remain closed. So that's called the hepatopancreatic sphincter. Another term for it, an older term, is called sphincter of Oddi. All right, and I think we are done. Let's see. Oh yes, histology of the pancreas. Okay, so you are going to be responsible for recognizing pancreas, which I kind of think looks like leopard print. And so if you think that too, just anything that looks like leopard print, you want to say it's pancreas. So let's check out what's happening here. So just overview of the pancreas. Most of this pancreas looks like this, right? These circular structures here, those are going to be the acini. Those are the cells that are pictured here, and those are making pancreatic juice. Those are the digestive exocrine. You can actually see the cracks leading to these ducts. Then we have these different looking population of cells, these pancreatic islets. This is where glucagon and insulin are being made. So these are endocrine cells, okay? So we have two different cells. So you're going to want to be able to name basically these two things. You're going to want to, number one, recognize this is pancreas, and then number two, recognize these are pancreatic acini, and then locate a pancreatic islet. And that's it, okay? So this is another image of a pancreas. It's different, right? But it's, you have a lot of these pink cells. They're kind of oriented in circles, and then you have a very different population of islet cells here. Okay. And this is one of my favorite cartoons of the semester. Mr. Turd hated driving his wife anywhere because she says this. All right. That's it.